As promised, we're continuing on with our topic of production of urine, looking at each step as we progress through the nephron. The first step we have to look at is the filtration portion, the part where we actually get blood filtered at the glomerulus, producing our filtrate that then continues on down the remainder of the nephron. Now, the big topics that we have to get out of the way when we're talking about filtration is understanding, number one, what drives this filtration, and number two, how we regulate it. Remember, we're talking about blood entering the afferent arteriole, solutes and water leaving through slits in the glomerulus into our Bowman's capsule, and then progressing down through the tubular portion of the nephron, including the proximal convoluted tubule, loop of Henle, and distal convoluted tubule. Any blood that does not get filtered at the glomerulus, and we're talking some water, formed elements like cells, and we're talking about proteins, are going to continue on out of the glomerulus through the efferent arteriole. So the big difference between blood in the afferent arteriole and the efferent arteriole is going to be volume and dissolved molecular solutes, with the only thing remaining in the blood by the time we're done with the efferent arteriole is going to be formed elements and proteins. Remember the orientation of our glomerulus. In through our afferent arteriole, out through our efferent arteriole. Of course, that's going to continue on into our peritubular capillary and vasa recta. And here is the actual filtration portion of our nephron, producing our tubular fluid containing water and the solutes that were originally dissolved in the blood. Just like we saw with fluid shift, Starling-Landis fluid shift at a capillary, we need to have some sort of hydrostatic pressure pushing fluid through the glomerular wall. Now, also just like we saw with Starling-Landis fluid shift, that force is going to be the blood pressure here. However, it's not just the gross blood pressure. We have to look at this as a modified blood pressure. Because while blood pressure does provide the forces that drive the filtration, directly speaking, we're going to call the forces at the glomerulus the filtration pressure. And the main reason we're going to make that distinction is because we don't want the filtration pressure to be directly correlated to the blood pressure. We want the blood pressure to provide the gross force that will drive and create the filtration pressure, but we want to be able to manipulate the filtration pressure so that it's not directly correlating with the blood pressure. Because in reality, we would normally see a relationship between the higher the pressure, the greater the filtration. That's what we see at a normal capillary. That's one of the factors that would come into play if we had edema, with that excess fluid leaving the capillary. But in reality, at the glomerulus, we don't see glomerular filtration rate change very much. We use the mechanisms of the glomerulus to take what the blood pressure gives us and create a localized filtration pressure that's somewhat independent of what the systemic blood pressure has to be. The only time we get a change in GFR is going to be during times of fight or flight. When the sympathetic nervous system takes over, we actually see a stoppage of blood going to the glomerulus. And of course, if we can't get blood to the glomerulus, we can't filter the blood. Therefore, we stop urine production. So now the big question comes, how can we manipulate the mechanism associated with the glomerulus to offset fluctuations in systemic blood pressure and give us a constant filtration pressure. We want to have a constant filtration pressure the majority of the time, and the only way we stay away from a constant filtration pressure is when we start getting fight or flight or sympathetic dominance. So we are going to look at the filtration pressure being what directly affects GFR. But because GFR won't change, we are going to use constriction and dilation of both glomerular arterioles to maintain a constant fixed filtration pressure, regardless of what the systemic blood pressure happens to be. We call this an autoregulation. So if we see a situation where systemic blood pressure gets too high, we should not see that reflect in a change in filtration at the glomerulus. Conversely, if we see a situation where systemic blood pressure gets too low, we should not see a change in filtration at the glomerulus. So by using our arterioles going into and out of the glomerulus, we can take the gross systemic blood pressure and use it plus the mechanisms of the arterioles to maintain a constant filtration pressure pushing fluid into the nephron. We are going to look at two different mechanisms by which we regulate these. They are both going to look at different aspects that would have caused GFR changes, but they're going to tackle them in two completely different ways. The first is myogenic. The myogenic is an arterial reflex based on local blood pressures. We will see the arterioles of the glomerulus constrict or dilate in certain combinations based directly on rises and falls in blood pressure being monitored at that location. The second mechanism we have is tubuloglomerular feedback. Tubuloglomerular feedback looks at changes in GFR that would have been secondary to blood pressure changes. And the mechanisms of our tubuloglomerular feedback work to counter those blood pressure changes. So already I want us looking at these as mechanisms that are in parallel and supporting each other. We have to stabilize GFR against changes in blood pressure. Myogenic regulation takes that blood pressure and we undergo changes locally to minimize the effects that that blood pressure would have had on our GFR. Tubuloglomerular feedback works to actually counter the blood pressure that had caused the problem in the first place. So they act on two different aspects of our scenario, but both have the outcome of stabilizing glomerular filtration rate when all is said and done. So myogenics, a direct constriction or dilation of the arterioles of the glomerulus in response to blood pressure changes. And of course, there's only two ways we can have blood pressure change. It can go up or it can go down. So we are looking for arterial changes to offset both of those variables. If arterial pressure is high, that is going to initially want to drive a higher rate of glomerular filtration. Just like we saw at a normal capillary with our Starling-Landis fluid shift, blood pressure goes up, more fluid leaves the capillary. So at the glomerulus, we're going to initially want to see that same phenomena occur. More pressure, greater force pushing fluid through the glomerular wall. When blood pressure goes up, smooth muscle of the afferent arteriole is going to constrict. Now, if you remember back when we did cardio, we had two variables that dictated blood flow to an organ: pressure and resistance. And if pressure goes up, we can also increase resistance to keep flow the same. Essentially, that's what we're looking for here. Pressure goes up, we constrict our afferent arteriole to increase the resistance proportionally. The higher the blood pressure, the more we constrict the afferent arteriole to give even greater resistance. So we're offsetting the rise in pressure by limiting the flow of blood. And by controlling the flow of blood to the glomerulus, we control how much blood gets filtered per minute. Therefore, we control the filtration rate. So blood pressure goes up, afferent arteriole constricts to limit blood flow to the glomerulus. Let's look at the flip side. Blood pressure goes down. Now we will see the afferent arterioles do the exact opposite. Still based on the same principle, though. Blood pressure goes down, afferent arteriole dilates to reduce the resistance. It makes blood easier to flow into the glomerulus. But in this case, just increasing the flow is not going to be good enough. We need a pressure to push that fluid across the capillary wall. Otherwise, we're just going to see blood go in through the afferent arteriole and out through the efferent. So now that we've restored blood flow to the glomerulus by dilating our afferent arteriole, now we're going to constrict the efferent arteriole. If we constrict the efferent arteriole, we make it more difficult for blood to leave the glomerulus. And what that does is it will cause a back pressure in the glomerulus, forcing more fluid through the glomerular wall than our blood pressure would have been able to accomplish. So blood pressure goes up, we constrict our afferent arteriole, and we offset the rise in pressure with an increase in resistance and a limitation of flow, keeping GFR constant. If blood pressure gets too low, we dilate our afferent arteriole to make sure we're still getting adequate volume of blood making it to the glomerulus, but then we constrict the efferent, creating a back pressure in the glomerulus. And essentially what we will see there is the filtration pressure at the glomerulus be higher than the systemic blood pressure, countering that drop in pressure that was limiting GFR to begin with, forcing a consistent amount of fluid. So we just saw both mechanisms now working in tandem to manipulate a filtration pressure and a blood flow at the glomerulus that will offset any changes in systemic blood pressure, thereby keeping a consistent glomerular filtration rate. In myogenic control, we didn't do anything about the systemic blood pressure. We were simply using our arterioles to counter that blood pressure's effect. If the blood pressure was low, it's going to stay low in spite of us using myogenic regulation to regulate GFR. If blood pressure is too high, it is going to stay high in spite of our myogenic regulation. This form of regulation does not go back and change the blood pressure. It simply takes what's given, manipulates the mechanism at the glomerulus, and creates a filtration pressure independent of the blood pressure. Tubuloglomerular feedback does change the blood pressure. Now remember, we looked at the relationship of the distal convoluted tubule with the glomerular structures, because that's the relationship we're going to look at to understand tubuloglomerular feedback. Because if you look at the name, tubulo, referring to the distal convoluted tubule, glomerular, referring to the glomerulus, we're going to use the contents of our distal convoluted tubule to dictate changes at the glomerulus. Again, in a means of stabilizing GFR. We detect the composition of the filtrate in the distal convoluted tubule by looking at our macula densa cells. They are sensory receptor cells detecting osmolarity of that distal convoluted tubule. We look at our juxtaglomerular cells responsible for producing the hormone renin. Renin has influence over our angiotensin cascade and therefore secretions of ADH and aldosterone. And we know they affect blood volume, and we know they affect blood pressure. So if our systemic blood pressure is too high, we will see tubuloglomerular feedback lower it. If we see blood pressure get too low, we will see tubuloglomerular feedback raise it up. So now let's take that and put it in context of our GFR again. If blood pressure is too high, tubuloglomerular feedback will counter and change the blood pressure, because that high blood pressure was going to try to increase glomerular filtration. So if blood pressure is too high, causing a trend of increasing filtration rate, our tubuloglomerular feedback lowers the blood pressure that was the problem to begin with. If blood pressure is too low, limiting our filtration rate, tubuloglomerular feedback and its mechanisms will raise blood pressure, countering the initial problem. So now we kind of have to go back and look at least a hypothetical, because remember, these changes in GFR aren't perfectly instantaneous. If GFR were to get elevated, if we had a higher than normal blood pressure and we start to see GFR increase, what we will see with that increased rate is more solutes crossing the glomerular wall. And knowing what we know now about that renal threshold for different solutes, if we get increased sodium enter our proximal convoluted tubule, we'll only be able to reabsorb so much. So we will see increased sodium enter our loop of Henle. That increased sodium at that point means even more than normal sodium will still make it to the distal convoluted tubule. And that's where we start to measure. If sodium is high at the distal convoluted tubule, we infer that glomerular filtration rate must have been elevated. High sodium in the distal convoluted tubule infers an elevated GFR. If the sodium concentrations in the distal convoluted tubule is low, we likewise, but conversely, infer that glomerular filtration rate must have been low. But remember, we always want the stabilized GFR. We don't want it changing unless it's fight or flight type scenarios. So with tubuloglomerular feedback, we see two things happening at once. We still see manipulation of the glomerular arterioles. If we see increased sodium at the distal convoluted tubule, we see the juxtaglomerular cells secrete ATP. We are going to see a decrease in the secretion of renin and erythropoietin. Remember, renin was involved in our angiotensin cascade. Renin is an enzyme that converts angiotensin to angiotensin 1. Angiotensin 1 gets converted into angiotensin 2 using our angiotensin converting enzyme. And it's angiotensin 2 that triggers the secretion of ADH and aldosterone. So, I've always thought that these scenarios were better now to look at in a flowchart manner. This flowchart, I have put both your myogenics and your tubuloglomerular feedbacks in the same chart. We just have to know where they are. Because remember, they're two different systems working together to stabilize GFR. Myogenics take the systemic blood pressure and accommodate to it to keep GFR independent of the blood pressure. The tubuloglomerular feedback more so make corrections to the blood pressure that was causing the problem to begin with. So here is our myogenics in red. Remember, they are a direct response to changes in blood pressure. Increased blood pressure, constriction of the afferent arteriole. Those were our myogenics. The rest of this diagram is tubuloglomerular feedback. When I say increase, when I say increase salt, that means increased salt at the distal convoluted tubule. So here in blue now, we will circle tubuloglomerular feedback. Increase salt at our distal convoluted tubule causes an increase in ATP to be secreted by our juxtaglomerular apparatus, which goes on to reinforce the constriction of the afferent arteriole. But it also decreases renin secretion, which limits the amount of angiotensin 1 present, which limits the amount of angiotensin 2 present. Therefore, we get less aldosterone, less ADH secreted, results in more urine being produced, less fluid getting brought back into the circulatory system, and we know lower blood volume can be used to control blood pressure. So we lower the blood volume, and therefore we wind up lowering the blood pressure that was a problem to start with. Because remember, we started with an increase in blood pressure. That was our main concern. Our myogenics accommodated to it extremely quickly. So it gives us some stabilization of GFR immediately. Tubuloglomerular feedback isn't so much worried about the immediate changes in glomerular filtration rate. But what the glomerular feedback will do is it will take the initial change in blood pressure that was the crux of the problem all along and work to correct it. So if blood pressure was too high, driving GFR up, tubuloglomerular feedback lowers it down. If blood pressure is too low, immediately we get our myogenics, constriction or dilation of the afferent arteriole, and we also talked about constriction of the efferent arteriole here. Remember, don't forget that one. Tubuloglomerular feedback looked at the sodium that was in our distal convoluted tubule, reinforced the changes at our glomerular arterioles. But if we see not enough salt or less salt than we expect to see in our distal convoluted tubule, we're going to increase our renin secretion. We follow that through the rest of our cascade. We will increase our ADH secretion. We will increase our aldosterone secretion. We'll increase our thirst reflex. Blood pressure will raise up because now we're raising up blood volume, which will take the initial change we saw in GFR and eliminate that blood pressure change that was the result. Blood pressure was decreased. GFR is going to take that blood pressure now and raise it back up as a way of stabilizing GFR. So we have to picture this as a negative feedback loop, right? If GFR is decreased, we release renin, angiotensin activation, increase blood volume, raising the blood pressure, and we normalize GFR once again. The only time we should see GFR change from this constant regulated state is during times of sympathetic dominance. Sympathetic nerves innervating our afferent arteriole will give us some of the strongest vasoconstriction we see anywhere in the body. It drives resistance to blood flow to such an extent that even the blood pressures created during that fight or flight response is not enough to push blood into the glomerulus. Therefore, no blood, no urine production. This is how all along when we said during fight or flight, urine production goes down. It's because of the vasoconstriction exerted on the afferent arteriole. If we can't get blood to the glomerulus, we can't filter that blood. So when we look at our key concepts now, it helps to study this in order. Understand how filtration occurs at the glomerulus to begin with. We have a hydrostatic pressure. And at that point, you can loosely call that blood pressure, pushing fluid out of the capillary and into the rest of the nephron. However, we have to quickly realize that because blood pressure can fluctuate, under that model, GFR can also fluctuate. But we want to keep GFR constant. So if the hydrostatic pressure of the blood pressure is what's driving that filtration, we need to make changes to our glomerular mechanism to accommodate and offset the changes that the fluctuating blood pressure would have created. If blood pressure goes high, we take our filtration pressure relative to it and drop it down. If blood pressure is too low, we take that filtration pressure relative to the blood pressure and raise it back up. So filtration pressure at the glomerulus stays constant in spite of a changing blood pressure. Take some time to digest this material, because this is an important feedback mechanism, not just for regulating urine production, but for regulating blood pressure. And if you feel like you understand this material, I encourage you to go on and read a little bit about blood pressure changes when we get a condition called renal stenosis. Renal stenosis is when we get a closing down of the large renal arteries going to the kidney as a whole. They get smaller. They limit blood going to the kidney. Through the same mechanisms we've just talked about here, the kidney is going to perceive that as a drop in systemic blood pressure that needs to be corrected. However, it's not a change in blood pressure. It's an increase in resistance. The kidney doesn't know that, so it's going to change its renin secretion and ultimately drive blood pressure up to overcome that resistance. And that's the reason why some of the highest blood pressures you will wind up seeing in a clinical situation will be those associated with the condition of renal stenosis. You get this vicious cycle going. That resistance of blood in the renal artery needs higher pressure to overcome it. And by the time you get enough blood flow going to the glomerulus, you are walking around with a resting blood pressure that's well in the 200 over 160 range. So as usual, if you have any questions, please let me know. Now that we have tackled filtration at the glomerulus and how we regulate it, now we move on to looking at each portion of the nephron and its relevant reabsorption and secretion at each place. We'll start with the proximal convoluted tubule and the loop of Henle first. Thank you.